Pedro.
Thank you, Nelly, for the very kind introduction. Hi, everyone. I cannot see you, but I'm very happy to be with you today. So, unfortunately, as mentioned, there seems to be a difficulty in sharing my full screen, so you will not get my animations, but I'll wave my Italian hands and hopefully make up for it. So, anyway, as Nelly has mentioned already, my lab has been focusing for many years now on colorectal cancer. Colorectal cancer is a tumor type that affects both women and men. You probably know this very well, but for those that perhaps are relatively new to this disease, here are the facts, and you can see that it's a very relevant cancer type. With respect to treatment, colorectal cancer has advanced like many other cancer types, but just not as much as we all wanted. And you can see the results of chemo and other regimens. Like I said, they are significant, but still a lot of work to be done. And in the last few years, we and others have introduced targeted therapies. I will discuss these briefly before going into the new data. I'll present a lot of unpublished work today. So a number of years ago, we became interested in targeting resistance to EGFR blockade. Antibodies against the EGFR are used in this setting, metastatic colorectal cancer. Resistance develops, and we reported a number of mechanisms of resistance, which are over there. They are now widely confirmed and even utilized in the clinic. And a few examples of what we have done for these is highlighted here. As Nelly mentioned, we have used liquid biopsy, for example, to track the evolution of these tumors in the blood. And here is what we found. We found, for example, that this is old data, 10 years ago data, but it's actually an interesting story because with very now primitive techniques to do liquid biopsy, we have observed what happens in the blood to the drop and rise of mutant alleles during the anti-EGFR monoclonal therapy. And you can see here that what happens is that when the therapy stops to work, there is a rise of resistance mutation. In this case, it's a RAS allele. I just showed this to prove that this has been repeated many times in many labs, and it appears to be widely spread. And what happens essentially is that you develop this resistance, and then we were the first to report that when the therapy is stopped, so we've stopped the therapy because there is clinical resistance, the allele that developed, the RAS mutation that developed, drops and reduces and goes back to almost zero. So, when you stop this selective pressure, something happens and the tumors becomes different. We use this clinically, and I'm very, very pleased to report the final results of Kronos, our first international interventional liquid biopsy study. Here we have used for the first time concept that came from preclinical studies to the clinical setting to target these tumors. And you can see the concept right here, and it's actually very simple. We monitor in the blood the rise of the resistance allele, in this case a RAS mutation, but in the last few years, we added other mutations as well, including EGFR extracellular domain mutation and other RAS pathway mutation. And when the therapy is stopped because of futility, we continue to monitor. When it goes to zero, we restart the therapy. And this is the story of Kronos. It's a positive paper, and most importantly, a positive study. And we submitted the story. It's with a big journal right now. We'll see how it goes. But we feel that this is important, mainly conceptually. Okay, of course, this is also pragmatically used now in the clinic. You restart this rechallenge therapy based on what you see in the blood, but also proves the point that you can follow cancer evolution and really help these patients. So we have a very strong number of patients that responded to the rechallenge therapy based on active monitoring in the blood. And you can see here the results. You can also see the downsides of it, right? I mean, all the patients that responded, unfortunately, relapsed. And so this tells you that the tumors really, once it becomes resistance, you can still rechallenge with the same therapy. And this way, we do this rationally in Kronos using the blood as a marker, but you can still, unfortunately, develop resistance. And when resistance develops, the mechanisms are multiple, heterogeneous, and very difficult to tackle further. And so, yeah, probably now 5 or more years ago, we started to think that we needed to change something, or we would still keep getting the same results, which is find a targeted therapy, apply it to the clinic, get some responses, but they were not durable. And so we sort of thought that these mutations that we discovered and others were reported as well, also neoantigens potentially, and so we could use them in a different way. And we were fascinated and started this journey by looking at a subset of colorectal cancer, mismatch repair deficient colorectal cancer. These are 15 to 20% in the early stages. They drop to 3 to 5% in the metastatic stage. This is because mismatch repair deficiency or alteration in genes that repair DNA are strongly prognostic. Not only they are strongly prognostic, but they, as everybody that is connected at this point knows, they are very predictive of response to immunotherapy. The reasons why they are predictive, I think, still remains a little bit of a black box, but certainly mutational burden, number of neoantigens, and other factors play a major role. And so, what can we learn from this mismatch repair deficient colorectal cancer? And what can we learn from neoantigens that develop from mutations? Well, this is a story that developed in my lab now 3 years ago. And essentially turning to a very, very interesting results, because what we did, we inactivated MLH1, one of the key factors that control mismatch repair in mammalian cells. And the story goes back to bacteria. These are mutase and mutel in the bacteria. They are rooted by the evolution to repair mismatch repairs. And you can see that we turn some cells that were mismatch repair proficient into deficient, and we injected these cells. These are all published work, so I go very fast. And we injected these cells either immunocompromised animals or immunocompetent animals, and we saw very different results. Essentially, these cells were rejected, basically. The same happened when we inactivated MLH1 in PDAC. The cells were also rejected, although the effect was less striking, but still very remarkable. And so next we ask, what are the functional bases? What do we learn from these? And what we have learned so far is that CD8 cells are key. If I suppress CD8 cells, then mismatch repair deficient cells are able to grow back in the mouse that is immunocompetent. We can do transplantation experiments whereby we first grow the tumor in an immunocompromised animal. I told you these cells will not grow otherwise. We transplant in immunocompetent, and you can see a delay in growth. You can also see that in this setting, we can test the efficacy of immunotherapy, and it's remarkably efficacious. So we have a model now, but we don't understand why it works. And of course, we can say mutational number, neoantigens, but what do we learn from this? Well, what we learn is that what it matters is that how long you inactivate mismatch repair for. So if you wait long enough, these cells will be generating a lot of mutations and likely be rejected. You can see this here. You can also get the same result if you clone the cells, okay? If you generate clonal neoantigens. So there are two major ways. One is to wait, and the second is to generate clonal neoantigens. So this will lead to rejection. But the next question is, why are the cells rejected? I mean, is this dependent on antigen presentation? And this came with a surprise. So what we decided to do is to take the very same cell I described to you before. So colorectal cancer cells, mouse-derived, in which we inactivated mismatch repair, took out MLH1. We superimpose on this inactivation of beta-2 microglobulin. So in other words, we impair the ability of the cells to present neoantigens. Okay, we did this effectively by inactivating beta-2 microglobulin. You can see the Western blot there. You can also see that we don't have major histocompatibility class 1 presented on the surface. And so the rest is published. We published last year, and essentially together with Luis Diaz, we were able to show that in this situation, it is a CD4 T cell dependent mechanism that rejects these cells. So in other words, what fascinates us is that the cells without beta-2 microglobulin are certainly able to grow back, but they are also still responding to immunotherapy, something not obvious. And why is that? Why do these tumors still respond to immune checkpoint blockade, despite the fact that they have a lot of mutations, but they cannot present them to the surface? Remains a little bit of a mystery, and certainly very fascinating. Importantly, human patients will do the same. So human mismatch repair deficient colorectal cancer and other mismatch repair deficient tumor types that have also beta-2 microglobulin loss, or in other mechanisms in which they have lost the ability to present the neoantigens, they, on the surface, they also respond to immune checkpoint blockade. All this suggested to us that perhaps we could do something crazy and unconventional. So perhaps we should try to inactivate mismatch repair therapeutically. So perhaps we should try to block mismatch repair in order to induce the responses that I just showed you. And so this is certainly a genetically validated pathway, has been confirmed in mouse and cancer patients. And you can see that the concept that is behind this is the fact that you inactivate the ability of the cells to repair DNA, and they become more suitable for immunosurveillance. Now I have a conflict here. I disclosed it at the beginning, my second slide. I repeat it here because we started a company around the idea to inactivate mismatch repair therapeutically. So basically what we decided to do is something that you probably would never think of doing, which is develop molecules that will bind mismatch repair protein and inhibit them. Now this is a long journey, still in the beginning, but we have molecules now, and we will see how it goes. We will see if these can turn into drugs. Perhaps they will have side effects. Perhaps it will be very challenging to use them. Perhaps, who knows? This is certainly a very, very high risk, potentially high pay project. So Neo4 is working on that. I'm very proud that the progress has been made. We made the crystals of the mismatch repair proteins. We identify inhibitors. It was more complicated as usual than we thought, but this class of proteins was never therapeutically drugged before. In the meantime, however, while we develop this drug, and who knows if they will ever work, how do we increase recognition of non-self antigens? Basically, how do we boost recognition of somatic mutations? Well, I present two ideas and the fallouts of these ideas around the concept that we should try to use neoantigens to improve immunosurveillance. Idea number one. Well, certainly there are a fraction of mismatch repair deficient tumors that respond to immunotherapy. But the vast majority of the MSS, mismatch repair proficient colorectal cancer, are very, very reluctant, recalcitrant to this therapy. And so, like many others, we keep thinking, how can we convert these tumors? How can we make them responsive? Or in other words, how can we turn them into hot cancers? And one idea came from observation from the clinic. There are patients that are mismatch repair proficient, so that the pathologist will report as mismatch repair proficient, in which, however, there are islands that are glands, that are portion of glands that are mismatch repair deficient. And we even observed this in xeno patients, so in mismatch repair proficient tumors that are transplanted in animals. You can see this patchy heterogeneous pattern of expression of mismatch repair proteins. The other reasoning that we did is that there are drugs that are out there already that are capable of selecting off mismatch repair deficient cells. Drugs like temozolomide, as shown here, or 6-guanine. And so we thought that perhaps we could use this concept to develop new clinical trials. And let me remind you what happens when an alkylating agent like temozolomide is given to a mammalian cell. The first thing that happens is it will alkylate DNA and will generate adducts that needs to be removed. The one pathway to do this is the MGMT-driven pathway, and basically this leads to ubiquitination. However, there are cells that are MGMT negative. Okay, when this happens, and there are cancers that are MGMT negative, it will get the cell into a futile cycle of trying to repair and fail, and eventually the cells that survive by doing mismatch repair deletion of mismatch repair genes, or otherwise inactivation of mismatch repair genes. Sometimes it's not deletion, in fact, it's epigenetic silencing. And we have proven this before, so we have shown that you can induce mismatch repair deficiency by selecting, for example, with temozolomide. And if you inject the cells that were selected with temozolomide, they are rejected by immunocompetent animals. And we have even had glimpses of this in human cells. You can see here a screening that we did in which we have treated with temozolomide to mismatch repair proficient cells, and we have observed that those that have MGMT negativity, they will develop resistance by inactivation of mismatch repair genes. This led to the Arethusa trial. The Arethusa trial is not completed yet, but we have some interesting results that I'd like to share with you. And this is totally unpublished and again, submitted to a journal. So the concept of Arethusa is the one that I just described to you. Can we essentially use drugs to select off mismatch repair deficient cells from a mismatch repair proficient tumor? And I already mentioned the two drugs that we were thinking of, temozolomide and 6-guanine. Now, the story that I'm going to tell you is actually much larger than the one I cramped into this talk because I was limited in time. But the idea is that we studied first what happens if we take mismatch repair deficient cells and mismatch repair proficient cells, isogenic, we mix them, and then we give them either 6-guanine or temozolomide. Well, you can see what happens if you mix 20 and 80% mismatch repair deficient and proficient, and then you select with temozolomide 61, you rapidly, in 8 days, pull out the cells that are deficient. But you can even mix 1% mismatch repair deficient cells and 99% proficient cells, and with these drugs, you will get a population that becomes highly homogeneous for being mismatch repair deficient. Now, of course, this is in vitro, but it's interesting enough to move it into in vivo. And if you do so, then the results are pretty exciting. In non-skin mice that have no very, very minor capability of performing immunosurveillance, you will see that these two drugs that are, the cells that were treated with these two drugs grow but poorly. But if you inject them in immunocompetent animals, actually they don't grow at all. Okay, so there is a profound difference here, suggesting that perhaps this is driven by temozolomide selection or 61 selection, which leads to immunosurveillance. Perhaps even more interestingly is these results where you can see really very profound difference in the ability of these cells then to grow back in either non-SCID or BABC, respectively immunocompromised or immunocompetent animals. Now, of course, the most interesting part of this is, can we do this in vivo? And so what we did here is to first inject the cells in the proportion that are mentioned on the left side of my screen. So 20-80, 50-50, and then give the mice, while the tumor is growing, 60-1 or temozolomide. But we use 60-1 because temozolomide in mice is actually, we found it is quite myelosuppressive and toxic. So we prefer to use 60-1. But the results are really remarkable here. As you can see, if I inject a population that is 50-50, and then I treat with either controlled DMSO or 60-1, the tumor initially grows, but then is rejected. And certainly that is not due to the ability of the ability of these drugs to suppress the growth of the cells, but rather to the immunosurveillance that is elicited by triggering selection of mismatch repair deficient cells, as you can see here. Now, one step forward, the Aretusa trial. So that gave us the idea that perhaps we should try to use this clinically. And so we developed the concept that is behind Aretusa. So Aretusa is actually a relatively complex clinical trial, but the initial results are very promising. And Nelly, please advise me when I have 10 minutes left, if you can, if you don't mind. Thank you. And so what is Aretusa? Aretusa is based on initial screening with MGMT. So we only want to enroll into Aretusa MGMT negative patients. That is already a lot of burden. And then we go into the priming phase. The priming phase is based on temozolomide. I just described you the concept that are behind it. And the next level is that those patients that in a post-temozolomide biopsy show increased mutational burden, or other aspects that I'll describe in a second, will go into Pembro. Okay, so the concept is we use priming with temozolomide to select off mismatch repair deficient cells, and then we come in with Pembro. And it's a complicated trial. The clinicians were fantastic and were able to pull this off largely during COVID. But as you can see here, we have several correlative studies, including collection of blood, a post-temozolomide mandatory biopsy, a pre-non-mandatory biopsy, and then the immunotherapy phase. And those patients that develop a mutational burden that is above 20 mutations per megabase, they will go into the Pembro phase. Now, what we learned from Aretusa is a lot preclinically and conceptually. First thing we learned is that we can use a signature to tell which patients have mismatch repair deficiency, or at least have inactivation of mismatch repair genes. Because we use a signature, sorry, I went too fast, that is related to temozolomide exposure. This has been reported by others. We use it, we adapt it to colorectal, we fine-tune it, and we were able to see that broadly the patients could be classified in 3 subtypes after the priming phase. The first subtype, which we call subtype 8, has nothing. There is no molecular evidence of temozolomide treatment. These are patients that receive very few cycles, that didn't respond to temozolomide, basically. Then there is a second type, which we call subtype B1, in which there is subclonal evidence of temozolomide treatment. Okay, and there is then subtype B2, in which we have clonal evidence of temozolomide treatment. And this evidence comes from mutational signature, which we computationally recognize as a very complex system in which we do all exome sequencing post-temozolomide. The biopsy is analyzed by whole exome sequencing, and we go on to measure mutational burden and mutational signatures. And you can see what I was telling you before. In Aretusa, what we realize is that to get at the temozolomide signature, you need to receive a certain number of temozolomide cycles, at least 4, basically. And you can see individual patients. If you expose the patient to few cycles of temozolomide, of course, because they cannot clinically tolerate or they don't respond at all, then they will not get the signature. We also learned that having a few pre and post biopsy, I told you that priming biopsy was not mandatory, but still we were able to pull it off in a few patients that consented to this. Then you can really compare now the various subtypes, and you can see the level of mutational burden that increases in subtype B1. We measure this in the tissue, and we measure this very, very carefully also in a larger number of patients, in which we measure clonally and subclonally the level of mutational signature. And you can see here that most of the patients that in group 2, actually we have only 2 now, possibly a 3rd patient, have a high subclonal level of temozolomide signature and a minor level of temozolomide clonal signature. We use blood as well. Nelly mentioned that we are very focused on liquid biopsy. So here is fascinating, because while we don't have always the pre-temozolomide tissue biopsy, we always have the pre-temozolomide liquid biopsy. And so you can see here blood mutational burden, how it increases during the temozolomide treatment, okay? You can clearly see that, especially the subclonal fraction, really increases, and we have reached statistical significance in this. What happens in the tumor? Well, what happens in the tumor, I told you there is this signature, which is alkylation driven, and the signature is actually driving a very specific alteration in one mismatch repair deficient genes in MSA6. And you can see the alteration right here. In most of the patients, we in fact are able to recognize almost invariably a specific mutation, but all the mutations that we find are actually linked to the temozolomide signature. And you can see here that others, luckily, this is the mutation that we find more often, but others were able to report before that these mutations are inactivating. So we know from functional study that the mutations that are occurring in MSA6 are inactivating. And you can see here that most of the patients, if not all of the patients, develop a very specific mutation. This mutation right here that is now used by liquid biopsy in our clinical trial to select the patients, or actually to identify the patients that are likely to have developed mismatch repair deficiency, because it occurs in 93% of the cases. And of course, this is not present before temozolomide, so it's temozolomide specific. Now you're asking, what happens to the pembro phase? And this is the most challenging part, because very few patients make it to that stage. But in a few instances, we have more patients now. We have had long responses. Certainly unexpected. This is a patient that responded for 24 months to pembro, and this is a confirmed mismatch repair proficient patient, so that developed this. And in fact, these patients right here. Sorry, go back. But these patients right here, the patient that developed this very high level of mutational burden, it's this patient here that had this remarkable response to pembrolizumab. Of course, too few patients, proof of concept, I totally get it. And temozolomide is not a great drug to dose in colorectal cancer, but we have also other data that we are collecting and additional correlative studies, including longitudinal liquid biopsy. And you can see what happens if you use this type by tracking the MSA6 mutation that I described to you before. You can see what happens during the priming phase and the treatment phase. So we're fascinated by these results. They conceptually lead to the idea that perhaps one day we could have drugs like these that will turn a cold into a hot tumor. How much time do I still have? Do I still have 5 minutes?
You have up until 12:50, Dr. Bardelli.
Oh, plenty of time. Okay, so I can tell you the final story then, but it's good to leave time for questions, and I don't want to bore you for too long. But I'll tell you the final story of today, which is story number 2, okay? I told you at the beginning that we were frustrated by getting resistance to targeted therapy, and we wanted to develop new concepts. And the concepts are around the idea that we could use combination of therapies to induce immunosurveillance. So idea number 2, it's actually out of the box a little bit idea, actually very out of the box idea, because what we are trying to use here to induce immunosurveillance is not a drug, it's a vitamin. It's actually vitamin C. Vitamin C is extremely controversial, okay? Has been used, and I'll describe in a second, in the cancer field for many years. But let's look at the facts. Vitamin C is a water-soluble intake vitamin, so we need to take it with the diet. We are not able to synthesize vitamin C. Mice are able to do so, by the way. And of course, it accumulates in leukocytes, is a cofactor for detected dioxygenase and histone demethylase. So there are a lot of beautiful work suggesting that vitamin C controls expression of genes, basically. And we certainly know what happens when we are not able to intake enough vitamin C with the food or with the diet in general. We have impaired immunity, we have severe diseases, which are not observed these days any longer. But certainly, if you don't intake vitamin C, there are deficiencies. And one important aspect is that cancer patients tend to have a low level of vitamin C in their plasma. Why did we stumble on vitamin C? A little bit of a sidetrack, but certainly related to colon cancer. So you know very well the group of Lou Cantley, and Lou and others, quite a few years ago, reported these very interesting results. Colorectal cancer that have either KRAS or BRAF mutation are very sensitive to vitamin C. And they had a mechanism. So we thought, oh, this is interesting. We should look into our very large collection of cells. We have around 270 CRC cell lines at this moment, including many derived from patients. And so, you know, let's go back and look at the literature on vitamin C before we embark into these studies. And you cannot do this if you don't go to this beautiful work of Linus Pauling, an incredible chemist, an incredible scientist. Not only that, but he won the Nobel Prize twice and one time for this work. And excitingly, Linus and Cameron came together at some point, converging on the concept that perhaps they could use vitamin C to treat terminally healed cancer patients. And so I recommend that you read these two PNAS papers that they published in the 70s. They were controversial, but certainly what they used was ascorbate, so vitamin C, in terminally healed cancer patients. And the results were, this was supposed to be one of the animation slide, but that's okay. And so in the 70s, I told you that they have used combination of vitamin C intravenously given. But then these results were repeated by the Mayo Clinic and were negative. So when they did a randomized study, these did not prove to be holding. And then there are other smaller or larger studies in which there was inconclusive results or benefit in a few patients, very inconclusive. Okay, this is the story of vitamin C. But the important thing is, Pauling insisted that he wanted to use IV vitamin C. Other trials, most of the time, have used oral vitamin C. Why is this so important? It is very important because if you use oral or intravenous, which in the mouse is intraperitoneous injection, you will get different levels of vitamin C in the blood. Okay, you can see right here. We have confirmed this in our mice, and as a result, there is currently lacking of evidence that the IV use of vitamin C is effective as a cancer treatment. However, you're probably also aware that in the US, especially, there are a lot of vitamin C clinics. You may be also aware that there are anecdotal reports of patients that have been discharged from major oncological hospitals going into palliative care at home that then are enrolling to vitamin C clinics and apparently have benefit from it. This was enough for us to become very interested. These together with some experiments, okay? And the experiments are right here. On the left-hand side, you have immunocompromised animals. On the right-hand side, you have immunocompetent animals. They have been injected with different mouse cancer cells: breast, colorectal, melanoma, pancreatic, the same, okay? And you can see here that they were treated with vitamin C intraperitoneous, and you can see that there is an efficacy of some sort only in the immunocompetent animals. These to me look pretty striking. I have seen many drugs that are very toxic that do this or little better or nothing at all. Some of these actually made it to the clinic. So if a vitamin does this in an immunocompetent animal, maybe we should study it more, especially because I have a control and the immunocompromised does not. We tested further the levels that we wanted to achieve, and you can see here that we want to achieve a high level of vitamin C to get an effect. In fact, we only need, it's not enough to give it orally. And then we studied a lot. We continue to study the impact of vitamin C, and you can see there some of the results here. So essentially, what we realized is that vitamin C will increase interferon gamma expression in CD4 and CD8 lymphocytes. So this is going beyond what Lou Cantley had reported. This is a non-cell autonomous impact of vitamin C, in addition to the possible cell autonomous impact. It's T-cell lymphocyte mediated. If you suppress T cells, the effect is minor or not at all. We have a very nice story that is developing on the impact of immunogenic cell death, vitamin C, and cancer, and restriction of cancer growth in immunocompetent animals. And certainly you can see some of these results, because if you combine the vitamin C treatment with immunotherapy, the results are pretty remarkable. And to be honest, I haven't seen these type of things before. And so we became excited. We are continuously trying to push a clinical trial. We may be able to pull this off finally. There's been a struggle because nobody really wants to give you vitamin C. Actually, we had to buy it ourselves and formulate, but that's a separate story. But the results are intriguing. You get infiltration. If you give vitamin C, you get a lot of other aspects that I don't have time to disclose today. But it's fascinating that perhaps at the end of the journey, and I hope I didn't bore you too much, we started off with targeted therapy, the concept of drug resistance, the shift towards new ideas on immunosurveillance. And I proposed to you two ideas. One is mismatch repair deficiency induction with drugs such as temozolomide or 6-guanine. It's actually more than induction, it's selection of mismatch repair deficient cells and immunosurveillance, or the idea that we could use a vitamin to increase immunosurveillance in patients. And if I want to tantalize you with the final results, if you use then the same model that I just described to you, so putting together mismatch repair deficiency and vitamin C, you get an amazing results, which is you cure mice. So essentially, mismatch repair deficient tumors that are injected and are allowed to grow to a very large volume. Look at here. We are treating when they reach about 1,000 or close to 1,000. Okay, most of the experiments currently are done in many groups when the tumors are very small. This is a very large mass, very difficult to control at this point. Typically, you will lose the mouse in very few days. But if you concomitantly give immunotherapy and vitamin C, you cure these mice. And these mice live happily thereafter. We kept them for a year. The tumor never came back. In closing, I try to convince you that there are several ways that perhaps we could use in the future to exploit the immune system together with chemo or other therapies to boost immunosurveillance. Some of it is cell autonomous, some of it is cell non-autonomous, so systemic essentially. And I've tried to convince you that there may be other ways that we can exploit heterogeneity and somatic mutation in cancer cells to the benefit of the patients. With that, I'd like to thank my group. This is how my group looked like before the pandemic. We are still unable to do a picture like this, but we are able to take pictures like this these days now. I'm just kidding. Italy is doing much better now, and we went for our first retreat finally with the entire lab. With that, I'd like to thank those that funded this research, from CRUK to Italian Association for Cancer Research, Fondazione Piemontese Ricerca Cancro, the European, the ERC grant that I just got, I'm very proud of it, and other grants that are listed here. With that, I'll stop sharing and be delighted to take any question if you have any.
Okay, thank you for you went through very fast, typical Italian
Sorry about
passion.
that. Yeah.
But anyway, let me get started with some questions until we may get some more from the audience. So I have a question first to the first part on the TMZ treatment, because you mentioned that, that would be an MMR deficient ones would be more sensitive, but it looks like if you're assessing the blood is wild type and you still see mutations, no? Am I misunderstanding something? So you do get mutations even in patients who are not mismatch repair deficient, because they see that in glioma also, that this is commonly used, so you will get mutations, so you should enhance immunotherapy.
We started off from mismatch repair proficient colorectal cancer, KRAS and BRAF mutant, so these are all NSS. We gave them temozolomide, and we don't have mutations before, we have mutation after, so it's pre and post confirmed. I'm not sure I got your question then. What is it exactly?
No, I just, maybe I just got lost because I thought you had engineered the cells to be MMR deficient and those were more sensitive, but anyway, okay. And then, but when you see the response to the TMZ treatment, do you see a difference in the location of the tumor? Mm-hmm. Which one is more sensitized versus not, like in terms of ascending or descending clonal?
Very difficult to address because we only have one post-temozolomide biopsy, which is typically taken from the most accessible lesion, and the blood, which of course will not tell us which one. I should go back and see whether some of the biopsy were taken, but these are metastatic patients, so I'm not sure I can address that question now.
Yeah, but I mean, in terms of where was the original primary tumor location,
well,
that could influence whether they respond or not.
That's a good point. I'm assuming you're referring to respond to temozolomide or not. We should go back and look at that. That we can do. That we can do.
Yeah. Okay. And then, so, you know, the vitamin C, I mean, you went through that part quickly, but is that, you know, the doses you use is very high. So it's actually, I mean, I see that one of the same question from the audience that I wanted to ask is that, you know, vitamin C at that high level could be actually an oxidizing agent because, as you know, everything is antioxidant or oxidant depending on a context as usual. So are you actually inducing oxidative damage by such a high dose of vitamin C?
We have, okay, so two ways to address this. We have done experiments in vitro in which we have buffered the oxidative power of vitamin C. And in that case, we still see the effect that I mentioned. The second way to address this is that we have performed adaptive transfer. So adaptive transfer, essentially we took mice that had no tumors, we gave them vitamin C, we took the T cells from these mice, gave it to other mice that had the tumor and saw an impact there, suggesting that at least part of the effect is not cell autonomous. However, like Luke Antlis showed, we do not rule out that part of the benefit is given by the pro-oxidant, for example, capabilities of ascorbate. Absolutely.
Okay, and it's only T cells that you need.
We have transferred, actually, we didn't do a very, very careful study. So we took the spleen. And so we are not sure, Nelly. So what we transferred was,
So treat the animal with the vitamin C and then transfer the spleen, and then that still acts the same way. Okay, interesting. But I mean, in principle, it could still
not the spleen, but the splenocytes. But that's what you mean.
Yeah. Okay. All right. So that was a question from Douglas Spitz. Is the ascorbate behaves as a pro-oxidant in cancer cells and an antioxidant in normal cells because of the differences in metabolism and superoxide and hydrogen peroxide and the ion. And does high dose ascorbate protect animals or humans against the normal tissue toxicity of immunotherapy? I mean, could this be used as a?
It's a good point.
Way to
no, to answer to that. It's a clever concept. The problem is, even if it is, I mean, it has been tested very widely, and so there was never a toxicity associated with vitamin C. Actually, not true. There is renal toxicity, but very rarely. So I guess you'll have to balance between giving IV for 3 days, for example, vitamin C, and protecting other organs if possible, with other risk. But I see where that's going, and it's actually an interesting point.
Yeah, but I mean, I guess if the effects directly on T cells and you may actually make them more active, it could be also making side effects even more worse.
That's also...
So it could go either way. So I don't know. But I have another question about the high dose, because as you know, ascorbate is also very acidic. So when do you see I mean, I know you people would be dead if you see a shift in pH, but do you see a shift in some pH in some local level in some cell types?
So vitamin C has been extensively dosed in humans, and there are plenty of studies. The maximum tolerated dose was never reached, basically, and it gets secreted very fast from the renal system. The only toxicity that we are aware of is renal toxicity, but it's actually minor. So basically, it is very difficult to reach a toxic dose of vitamin C, even in humans and even IVs.
Okay. And the iron, you know, that was related to the question, the iron concentrations, because, you know, many metabolic enzymes and other enzymes require iron as cofactor. So why the vitamin C may be affecting that?
How vitamin C actually benefit the immune system, I think, remains largely unstudied. And like I said, very briefly, and apologize if I went too fast, but we have a story on induction of immunogenic cell death. So the other possibility is that vitamin C creates a stress of some sort, in addition to activating the T cells, that will boost immunogenic recognition, essentially, which we have observed repeatedly, even with a lot of control. So that's also a very exciting twist. And I realize that is controversial, but at this point of my career, I don't mind to try something very controversial if it works, you know, because most of the other things appear to be working limitedly.
Yeah. There's a question by Gordon Freeman is, how is vitamin C metabolized? Any effects on metabolic pathways?
Yeah, like I said, it appears strange, but we went straight into vitamin C having read the literature, which is, you know, very large and very extensive. And I don't think that metabolism was ever shown to be affected significantly in patients from the studies that were conducted. So I don't think there is a major impact. Although what I think it is happening, there is major transcriptional rewiring, because that's what appears to be doing. So I cannot exclude that there will be a secondary impact on metabolism, essentially.
Okay. One more question by Emily Green is, she's also curious about the vitamin C story, and cancer-associated fibroblasts have been shown to play a role in both breast and colon cancers. Do you know what vitamin C does to the subtypes, you know, the cancer-associated fibroblasts, and particularly the subtypes? You know, there is some that's very high in LOX and all that. So
that's something that is actually one of the latter story that we are writing up. So the immunophenotype, essentially, of a tumor post vitamin C becomes really exciting, right? Because I alluded to the fact that if we suppress CD8 cells, we don't see the impact, but we have more recent data on NK cells, so for example, that play a role. And the way we approach these now is, you know, using the knowledge of the immunologist and the Fortessa, for example, to do a very accurate immunophenotyping. We did go to the level of studying macrophages. We haven't yet looked at the impact on CAF, but we are absolutely very intrigued by the rewiring of the environment. So that's one of the things that the postdoc is doing.
Okay. Okay, sounds good. Thank you. So I think, you know, we answered all the questions we had in the audience. Thanks, Alberto, again for the great talk, and looking forward to all these papers that seem to be cooking. And thank you, everyone, again for joining us today and listening to the seminar. And sorry about the screen sharing issues that we had. So thank you.
Thank you all for inviting me and for having me today. Thank you.
Yeah, thanks.
Ciao.
Bye-bye.
Bye.
Bye-bye.